All right, so thank you for that, incredibly interesting. Our next presenter is Elif Sikoglu, who's a postdoctoral associate at the Center for Comparative Neuroimaging at the University of Massachusetts Medical School at Worcester. Elif's been working in a variety of different therapeutic areas, psychiatry areas, but has been really focused on work around proton magnetic resonance spectroscopy, which is a very interesting emerging technique, and she's going to present some of her work in this area. Elif? You got this? Okay, and you've got this. I'll let you know yellow when you got 5 minutes, okay?
Thank you, and thanks to Autism Consortium for giving us an opportunity to share our work with you today. I'll be talking about glutamate and Gaba function in autism spectrum disorders. This work has been done in University of Massachusetts Medical School, and it's a joint effort between Center for Comparative Neuroimaging and Child and Adolescent Neurodevelopment Initiative. Understanding the clinical importance of glutamate and Gaba in autism spectrum disorders begins with the elegant work that has been done in the recent years on the molecular mechanisms underlying Fragile X syndrome. Fragile X is the leading genetic cause of autism, accounting for approximately 5% of the cases in autism spectrum disorder. The FMRP protein, as mentioned earlier today, is involved in the inhibition of the neuronal protein synthesis. When FMRP is not functional, overactivation of glutamate receptor results in overabundance of protein synthesis, resulting with the characteristics of Fragile X syndrome. And new treatments have been targeting this mechanism, relying on either glutamate receptor antagonists for glutamate receptors or agonists for GABA receptors. The work in Fragile X has been extended in larger emerging story that implicates an imbalance between the inhibitory GABA and excitatory glutamate. In this work that I'm presenting today, we aim to acquire glutamate and GABA levels in vivo and correlate those with behavioral and clinical assessments. To this end, we had 8 patients with autism spectrum disorders and 10 typically developing healthy controls. Both groups were composed of males, and they were age-matched. And the patients had the DSM-4 criteria confirmed diagnosis with IQ greater than 70. And the typically developing subjects had no prior history of psychiatric disorders. We performed a number of clinical assessments on these patients and subjects, including the IQ test, reading the mind in the eyes test, social responsiveness scale, social communication questionnaire, aberrant behavior checklist, and CUI box. Very briefly, in the reading the mind in the eye test, for those of you who are not familiar, participants are asked to match the pair of eyes with the best descriptive words. This test has 2 different versions, a child version and an adult version. Due to the age range of our interest, we administered both versions to come up with a combined score. The social responsiveness scale is a 65-item rating scale that measures the severity of autism spectrum symptoms, and it has different subsections that we conducted. Then we pursued an imaging experiment using an MR system. We collected GABA and glutamate measures using magnetic resonance spectroscopy. I want to underline that this is a non-invasive method to quantify the brain metabolites in vivo. And we used different pulse sequences. For both pulse sequences, the region that we were interested in, the region that we acquired data from, was anterior cingulate cortex, outlined with the green box. We concentrated on this region because it's a crucial area involved in limbic system. It's involved in planning, self-monitoring, and social interactions. From the first sequence, we could get glutamate, the GLU peak, and the glutamine, GLM peak. However, to get GABA, we had to use some edited pulse sequences. Again, for GABA, we were interested in the same region, anterior cingulate cortex. I'll cut to the chase and present our results. Both groups were age-matched, as I mentioned. However, there was a difference in the IQs. This is unfortunately a confounding factor in our study, and when we pursue this line of work, we will factor in for this. For the behavioral and clinical assessments, two groups were significantly different than each other. The patients performed worse in the reading the mind, in the ICE test, and the social responsiveness scale. Our MRS results revealed for glutamate and glutamine peak, the combined peak, there was no statistical significance difference between two groups, and same was true for glutamate. However, glutamine, the patients had higher levels. We must underline that glutamine is a storage buffer for glutamate. Glutamate is taken up by the astrocytes and converted into glutamine and shuffled back to neurons for processing back to glutamate. So what happened with GABA? Although the difference wasn't statistically significant, the patients had lower GABA levels, and GABA is synthesized from glutamate within the neurons. So these results kind of suggest that the excess amount of glutamate is converted into glutamine instead of GABA, contributing to this imbalance between these metabolites. We correlated our MRS findings with the behavioral measurements and showed that glutamine was inversely correlated with our clinical assessments. For example, in the reading the mind in the eye test, higher levels of glutamine corresponded to lower scores, which was worse behavioral. Same was true for social responsiveness task. So I want to end with posing few questions that we are looking into. So can we use glutamine and GABA as a biomarker for social cognitive deficits? Of course, we need to extend this work to a larger population, and within this population, we are planning to account for the age, gender, and IQ effects. Also, the next question is, we collected data from this anterior cingulate cortex, but what are the other key regions that are possibly involved, such as amygdala or hippocampus, and how do these metabolite levels correspond or correlate with the neural circuits? And the last question is, can we use these biomarkers that we are talking about as treatment response predictors? I would like to thank all the members of Candy and CCNI Lab, especially Constance Moore for leading the imaging part of the work, and Dr. Jean Fraser for leading the clinical aspects. We have a few disclosures. And thank you.